Well, first, I'd like to thank the organizers for giving me this opportunity to tell you about the work we do. But I have to admit, this morning, I wasn't feeling so good. And the reason was this, is that the projects that were being described were really, huge genomics projects, really big scale, asking big questions and applying the power of genomics. And, we do okay work, but we don't do this large scale kind of genomics kind of science. We're sort of, in that regard, we're sort of like the mom and pop store compared to the multinational corporations like this morning. But I thought, well, I'm trying to look for the positive and thinking, well, contrast is good, all right? There's, in piano concertos, the first movement is usually really big and really bold and highly technical. The second movement is a little more subdued to get you ready for the third. So we're in the second movement right now, short interlude. Hopefully you'll find some things that are interesting here. So we study cold acclimation. Why? Well, from the introduction, I was in Los Angeles for many years. Then I moved to the Midwest, and it's cold in the Midwest. And I said, looking at those plants, I go, how the heck do they deal with this? And so I decided to work on that, and I wanted to learn about that. And there's also the practical reason, the application side of that. And as we all know, we're faced with major challenges over the next decades to provide enough food and to provide the energy that we need as peoples and as societies. And biomass is part of that, solving those problems. Of course, and it turns out that abiotic stresses are a major limiting factor. They limit where you can grow various crop species. They limit and where you do grow those crop species. On an annual basis, they cause big losses. We happen to be interested in freezing tolerance, but there's some connection with these other cross protection, like with drought, which I'll briefly mention. So, as we all know, plants differ markedly in their ability to deal with freezing temperatures. Tomato has essentially no freezing tolerance, whereas wheat and rye have a considerable amount of freezing tolerance. But what's important to appreciate is that that's not a constant property of the plants. If you grow tomato and wheat at warm temperatures, at the same temperatures, there's not that much difference in their freezing tolerance. There is some, but there's not that much. What wheat and rye can do that tomato cannot do is sense the low temperature and activate mechanisms that lead to an enhancement of freezing tolerance. And that process is known as cold acclimation. And this is an example of that with Arabidopsis, which is the plant we do most of our work with. So if you grow Arabidopsis at warm temperature, 22°C, and then you just freeze them to -5 and then look what happens, well, they don't survive. If, however, you take those plants from warm temperature and you move them to cold temperatures for a few days, then freeze them, they survive. So the big question over the years has been, what's occurring in response to low temperature treatment that's leading to this enhancement of freezing tolerance? And when we got involved in this a number of years ago, we had a very simple hypothesis, and that was in response to low temperature that there were probably changes in gene expression that were occurring, and at least some of that was conditioning this enhancement of freezing tolerance. So we set out to test that hypothesis, and the bottom line is that indeed there are changes in gene expression, and they contribute to an increase in freezing tolerance. So what I'd like to do is give you a bit of that story. I want to talk first about things downstream from this first. When you put them at low temperature, you unleash this regulatory program that ends up resulting in freezing tolerance. So I want to talk about that. We want to know the genes that are being regulated by low temperature. We want to know the transcription factors that are controlling them. We want to take these changes in gene expression and organize them into regulons and understand their functions. And then we also want to know what's upstream of this first wave. How is the plant sensing the low temperature? What's the nature of the low temperature thermometer? And how does this all set off? And over the course of a number of years, what we did, we described what we call the CBF Cold-Response Pathway. The Shinozakis came at this same pathway through drought, as it turns out. So the hallmark characteristics of this pathway are that the plants are exposed to low temperature. These three genes, CBF1, 2, and 3, they're induced. They happen to encode these transcription factors, C-repeat binding factors. They bind to this regulatory element, which is out in front of about 100 or so genes. When these genes are expressed, it leads to this enhancement of freezing tolerance. And there are many mechanisms that are involved there. Some of the work we've described showed membrane stabilizing proteins. There's the production of cryoprotectants. I won't have time to go into that story. But let me just show you a couple of pieces of data to substantiate what I'm saying about this pathway having a role in freezing tolerance. So if these 3 CBF genes, they happen to be located right next to each other in the genome. You put the plants at low temperature, they quickly come up in response to low temperature. These are northerns over here. The CBF proteins are made, they sit down, the promoters are reported, their target genes, and you can see then there's this wave of changes in gene expression. So then the question would be, what would happen if you took these genes here, these CBF genes, and expressed them at warm temperature? Would these target genes be expressed? And the answer is yes, indeed they are. You can make transgenic plants that overexpress CBF, 1, 2, or 3. And even though they're non-acclimated, they're at warm temperature, the target genes are being expressed. Okay, so then the question is what's happening to the freezing tolerance of those plants? Well, it turns out, whereas these are not freezing tolerant, these are freezing tolerant. Interestingly, it's also true that they are more drought tolerant. And the Shinozakis were actually the first to publish on that. You might say, what's the connection here between freezing and this drought tolerance? Well, it turns out the primary damage that's caused with freezing, the injury is resulting from the dehydration stress that's occurring with the freezing. Okay, so to be freezing tolerant, you have to be tolerant to dehydration stress, and therein lies the cross protection. Okay, so that's in Arabidopsis. If one has the hope that ultimately one can use this information to improve crops, it would be nice that this pathway was outside of just Arabidopsis, and in fact that's the case. These are really early on studies showing, like in canola, rye, and wheat, there are CBF genes. They're induced quickly in response to low temperature. There are the equivalent, these orthologs of the core genes in Arabidopsis. They are induced. Secondly, if you overexpress CBF in canola, you have an increase in freezing tolerance. If you also you get an enhancement of drought tolerance. Okay, and this is not just in our lab. This has been seen in many, many labs. So if that's the case, then don't we have a bunch of freezing tolerant, drought tolerant plants, crops out there right now? Okay. Well, as it turns out, if you just express the CBF genes in a certain way, the plants really think they're cold. Their metabolism is actually, they have tenfold higher sugars. They convert over to this low temperature sort of physiology. Anyway, the bottom line is that if you just constitutively express it, you have significant effects on growth and development. You get these smaller plants here. You have much less yield, not a particularly good agronomic trait. And you also get changes in architecture, which is pretty interesting. You notice this flat stature right here. That's actually the more freezing tolerant plants. They lie flat to the ground so that they hope for snow cover to insulate them from the colder air temperatures. So it's actually a mechanism that's involved in freezing tolerance. So of course, what people are trying to do is use inducible promoters to turn this pathway on, and stress-inducible promoters met with some mixed success. There aren't products out there at this point. On the other hand, in certain circumstances, you can take a hit in yield. Let's say it's either you get to grow the plant or you don't get to grow the plant. In those kind of cases, you might have an application. And for example, with Arbigen, Maud Hinchey gave me these slides that I could show. They're interested in this tropical eucalyptus. It has properties for biomass that are quite positive. The problem is, in the United States, at least where they grow their products or their trees, it can only grow down in here, and it can't grow up in here. Okay, so they have CBF overexpressing plants with inducible promoters, stress-inducible promoters, and this is from a couple of this is from flying over this site over here, and you can see here that these are the transgenics that are surviving. The other ones, the wild type, did not. So there are potentially those kinds of applications. All right, so that's a CBF pathway, but a basic question is, to what extent then are the changes in gene expression that are occurring in response to low temperature are due to the CBF pathway, or are there other major factors important as well? And how much of freezing tolerance is dependent upon it? This is an early study where we're using an 8K array, then we use a full genome array. But the point was that you could see when you put the plants at low temperature, you get these waves of changes in gene expression affecting hundreds of genes going up, hundreds of genes going down. And the question is, how is all that then wired together? How much of that is CBF controlled? Well, this is some fairly recent examples of these kind of data. Here's from Colleen's work here. These are just replicate samples, okay, grown at warm temperature, cold temperature. Here are them at cold temperature. And heat map, and you can see this is at warm temperature, this is cold. They're increasing in response to low temperature. This over here are warm-grown plants overexpressing CBF. And what I think you can see is that these genes are not very much changed in response to low temperature, CBF expression, whereas these are like in the cold. Okay, so about 20% of the genes fall under the CBF Regulon. That's assuming, of course, CBF is in itself sufficient to turn on the genes. That's a big limitation here. So one of the things, of course, we'd like to do is take CBF out of the system and see what happens to cold acclimation. Does it completely crash? What happens to changes in gene expression at low temperature? Unfortunately, CBF 1, 2, and 3 are right next to each other, so you can't get individual knockout mutations and recombine them in. But we have a tool that came out of a suppressor screen for something else that is giving us some insight. This particular version of CBF2 only has the DNA binding motif but doesn't have the activation domain. So the idea is if you express this at high levels, it will bind to this sequence here, will not activate gene expression. And then if you put the plants at low temperature, the endogenous CBF proteins cannot gain access to the promoter. So it has a dominant negative effect. Here's one of the CBF targets, and you can see in three different transgenic lines, the induction of this gene is down 80 to 95%. This over here, raffinose, is a cryoprotectant that falls under the CBF Regulon, and this GAL-S3 is the rate-limiting step. So look, in the wild type, in the warm, there's little raffinose. It goes up in the cold. Then over here in these dominant negative plants, there's very little. Okay, so that's with one gene. What's happening at a genome level? Well, you can see that here, kind of. This first lane over here, these are all these cold-regulated genes. This shows CBF expression, the genes that are on in warm-grown plants. These are cold-treated plants that are expressing this dominant negative version of CBF2. So if there was no changes in gene expression, if it was having no effect, this lane right here would be all black, okay, because it would be the same as this. We're looking at differences to this. And what you do see, not very well, unfortunately, here, but there are these green bars that are over here for downregulation, and those are the genes that are normally upregulated by CBF. So it looks like we're having a significant effect of downregulating genes that are controlled by CBF. Although you can also notice there's all sorts of genes up here that are not still under the CBF Regulon. So what about the freezing tolerance of these plants? Well, it turns out they can still cold acclimate Here, non-acclimated dominant negative guys, cold acclimated ones, they're still increasing, but the degree to which they increase in freezing tolerance is not as great as in the wild type. So you can see here, right here, this is freezing to various temperatures below zero. This is electrolyte leakage to assess as a readout for cell damage. These are non-acclimated plants, the wild type and the CBF2 dominant negative. This is the wild type, this is the dominant negative. But you still get quite a bit of enhancement of freezing tolerance. So that says where are the other transcription factors that are important here? Well, this isn't going to, as it turns out, oh my, as it turns out, this is not going to be an easy, it's not going to be easy to figure this thing out because of this. Okay, these are the changes in gene expression, hundreds going up and down. Just look at the ones that are going up. Within this, there are 174 transcription factors. Okay, they're going up. And you notice they're going out they're rolling out in a developmental program. So one of the things we're trying to understand is just the regulatory logic used among these that are having those transcription factors roll out with that kind of regulation. We got CBF initially about a one hybrid screen, and so what we decided to do it was on a bigger scale and with a modification out of Walhut's lab. So basically what we did was this. We fused, in this case, we've actually fused 1,000 transcription factor open reading frames to the activation domains, put them into yeast strains, but anyway, here are the cold regulated ones here. We have whole promoters. That's the advance, one of the advances from this, that you can actually use whole promoters instead of just individual elements. And you mate these together and you see if this can bind to this element, you can turn on the reporter gene. We looked at 26,000 interactions, and the protein-DNA interaction map that we get is really quite complex, as you can see right there. These are interactions of the different transcription factors with each other. One might have hoped that the ones that, let's say here, the ones that are turned on here, that they would have bound to the promoters here, and those would have bound to the promoters here, would have bound to something like simple like that. Not that way. Much more complex. But there is an interesting thing that comes out of this. If you look at all of those interactions, these are transcription factors here that are binding to transcription factors here, and these are ones that are binding to the promoters of those transcription factors. But the point is, it looks like this thing might be a regulatory hub, and that one, if you look at this is another way to describe that, if you look at the genes that it's being affected in the promoters, it turns out that there's an enrichment for this sequence, which happens to be the consensus for a homeodomain protein, and this happens to be a homeodomain protein. So we're encouraged to think that these are actual targets. And in fact, we've only looked at a few of these now, but in a knockout mutation or downregulation, what we see are some of these putative targets are now not expressed at high levels. This one's knocked down, say, 50%. This is in the cold in the wild type and the mutant. Here's wild type and the mutant. So, and we're testing for freezing tolerance now. The other approach we're using is how we got to CBF initially to overexpress these. We just chose other transcription factors that were being expressed along with CBF. And so about 15 of these. And it turns out, again, you see a very complex interaction. So these are the ones that we express. These are the genes that, this is just then we do microarrays and then we look at the genes that are being affected. And so some of these transcription factors, like CBF, affect a lot of genes. Some affect a very small number of genes. Some, like CBF, remember I said CBF might be interacting with other transcription factors? Well, this ring right here are genes that are affected by at least 2 of the transcription factors. These right here are genes that are affected by 3 or more of the transcription factors. So very complex interaction. That's an insight that I hadn't necessarily been prepared for. We didn't know what the answer was going to be. It's pretty complex, and where we're now, one of the interesting findings is that you look at the freezing tolerance of all these, only CBF is the one that's giving us a big change in freezing tolerance. This one, ZAT12, gives us a little bit, and the rest of them don't do anything. But that could be for a number of, that makes you think that, okay, well, cold acclimation, yes, there's freezing tolerance, but there are other phenotypes that are associated with cold acclimation, like changes in architecture, like changes in photosynthesis. And so those may be things that are being controlled by these other transcription factors, and those are the kinds of studies we're into now. In the few, do I have like 5 minutes? 5 minutes? Okay. In terms of moving upstream from here, 5 minutes. When you look at these genes that are, these transcription factors that are all induced early, this first wave, an interesting observation is that they are all induced also by mechanical stress. They're all induced by treatment with cyclohexamide. That makes you think that these are wired into some central kind of sensing system. At least it makes me think that. Okay. So we're trying to understand what that sensing system is. We're working our way through the CBF promoters. And this just shows that the promoters, we defined sequences that were imported in imparting cold-regulated gene expression. And one of these sequences, we call these things ICE for inducer of CBF expression. One of these sequences overlaps this thing. Okay, well, that's interesting. Why is that interesting? That's the recognition sequence for a family of transcription factors called CAMTA proteins, CAMTA transcription factors. There are 6 of them in Arabidopsis. Well, why is that interesting? Well, these interact with Calmodulin, which is a calcium sensor. So why is that interesting? Well, 20 years ago, Mark Knight showed that when you take plants and you either mechanically agitate them, like over here, you get spikes of calcium. Interesting about that mechanical agitation. I just showed you about that first wave. Or if you take them from 20 and put them at different temperatures, some cases you don't get any spikes, but you go to low temperatures where you get induction of CBF, you get these calcium spikes. And there have been pharmacological studies to suggest that that was important in the regulation of some of the cold-regulated genes, but there was no link. Well, it turns out that this sequence by itself, if you hook it up to a reporter, it's cold regulatable. Interestingly, that same sequence will give mechanical regulation, give cyclohexamide regulation. So all that stuff's coming down onto this element right here. And what I'll do, because of lack of time, what I can tell you is that one of those transcription factors, CAMTA3, is involved in sitting at that locus and turning on. It's important in the activity of that particular CAMTA binding site. Okay. Also, if you knock out CAMTA1 and CAMTA3, you have an effect on, and I can't go through the detail, but on freezing tolerance. Okay, so we're starting to make progress here. It looks like at least one of these pathways involving this calcium going through calcium sensors through the CAMTAs to turn on CBF and other genes. And in 2 more minutes, I just want to give just one final thing because I think it's really cool. The clock. This has to do with some of the questions that were raised earlier today about circadian rhythms and stuff. Okay, Stacy Harmer a number of years ago showed that CBFs are subject to circadian regulation. Okay, they go like this during 9 days. And it's a circadian. But they're really low levels. Sarah Fowler, postdoc in the lab, she asked, well, what would happen if at this point of the day I dropped the temperature, and what happened at this time? Would it different in terms of cold induction? Well, indeed, if you drop the temperature at this point, big induction, drop the temperature at that point, low-level induction. Okay? So is that really, though, circadian? This is, you're dropping it in the day, this you're dropping at night. Maybe it's a diurnal effect. Well, if it's a circadian rhythm, what will happen is you go day, night, day, night, day, night, and you just keep them under constant light, and you'll still see the rhythm. And indeed, that's what you see. Day, night, keep them in constant light. But this is where the night would have been, and again, up, down, up, down. So the big question has been, what is important in the circadian regulation and what is important in that gating phenomenon? Well, it turns out that if you look, the circadian loops are complicated negative feedback loops. This core loop happens to have two players called LHY and CCA1. They can be positive or negative regulators. And it turns out they bind to a thing called an evening element. If you look in the CBF locus, there are lots of these evening elements. If you do chromatin immunoprecipitation experiments, you can find, these are mock experiments, these are real with the antibody to the protein, you can find it loaded up with CCA1 and LHY. And again, just to go, so the question is, if you knock out CCA1 and LHY, what happens in terms of circadian regulation? Just look at this one. This is the wild type, this is the mutant, knocked out. What about cold regulation? Here's the wild type, this mutant's knocked out. What about freezing? In non-acclimated plants, there's a significant reduction, about 50% loses the freezing tolerance here. And there is even some effect in cold acclimated as well. And so basically, and this is the last slide, in the daytime, under warm temperatures, you have CCA1 and LHY here activating, and then here, if you put them in the cold, you have CAMTA and other positive regulators. So you have this integration of the low temperature and circadian regulatory pathways. And as I showed, that it looks like that integration has an effect on overall freezing tolerance. It might have been an important evolutionary event to allow plants to move into colder regions. So that's what we got there. I'm sorry for running long. Our funding, these are the people that did the work. Sarah Genwine, my project manager, lab manager, 2 graduate students, 2 postdocs, and technical help, and funding source. Sorry for running long. Yes.
I was just wondering if you looked at any crosstalk between the CBF pathway and vernalization response?
Yeah, so vernalization response is a long-term cold exposure, involves epigenetic changes in the chromosome. It's a longer-term response. It's one that's designed for the plant. It controls flowering, and it's designed so that the plant knows it goes through winter. Hey, I've been through winter. When it gets warm, that means it's spring, and it can go ahead and flower at that point. This cold acclimation is more of a quicker response kind of pathway.